out of beekeeping completely because I could not keep my bees alive. And it was when I started putting things together. I tell people beekeeping is not one thing. It's not controlling Varroa. It's not your feeding program. It's not how you manage the colony, you know, for disease and whatnot. It's everything together. So what I'm going to talk about here is the Varroa aspect, because this one to me is the most important one. Anybody here anybody in this room that kept bees before 1988? Okay, there, you're not old enough, but he remembers, John remembers, right? Before Varroa, easy to keep bees, right? You put your bees in the springtime, you came and took honey at the end of the season, you closed them up, you came back in the spring and they were still there. It's not that way anymore. So I wanted, you know, this talk is going to be pretty general about as much about Varroa as I can tell you. And just to give you a little bit of biology and science behind it, because the more you understand how Varroa work, the more successful you'll be in the management of your bees. Okay? So, what is it? That's a nice pretty picture of a Varroa mite up close and personal, right? We have those on our bees. Do you have Varroa in your hive? Anybody here that has no Varroa in their hive? You have to leave the talk if you don't. I know he doesn't. Jeff never has any. But we all have Varroa. We will never get rid of it. We always have, no matter what treatment you do, there will always be some mites in your hive. So the key to this is controlling them. We have them. Is it going to be a problem? Maybe. Maybe not. But if you do nothing, it absolutely will be. So I want to talk a little bit about biology of Varroa mites, because I think the more you understand as a beekeeper how this all works, the more you can tailor your management program around Varroa and prevent maybe a blow-up of mites in your hive. Varroa mites have 8 legs, and they're a member of the arachnid family. They're highly destructive to honeybees, the Apis mellifera. The Asian honeybee, which grew up with Varroa mites, can tolerate them. It's almost, you know, they can tolerate the mites to a certain point, and they don't die from them like ours do. Our bees were never exposed to mites until, like I said, we brought them over from Asia in the, I think it was the late 80s, and they just wiped out our colonies. People that are my age or older, or maybe slightly younger, remember the days of seeing the bee trees everywhere, in the barns. You'd see the honeybees coming out of the barns, in the siding, anything with no insulation, with that cavity, there'd be a feral colony in there. Now it's very rare to see a feral colony, right? And any colony you see that's a feral colony today is typically a swarm from somebody's hive. Honeybees will collapse normally an untreated colony will collapse within 3 years. So we just don't have those colonies anymore. I get people telling me that, oh, we've had bees in that tree for 30 years. Well, maybe so, but it's not the same colony. That colony probably died over winter and a new swarm moved in. But, so they're highly destructive. That second point up there is incorrect. It said they feed on the hemolymph of the bees. That's what was believed up until this past year. Dennis Van Engelsdorp and his team was doing a lot of research, and they found out that they're really feeding on the fat cells in the honeybees. So it's not the hemolymph. And that actually makes a lot more sense, because we're seeing our bees collapse from mites in the wintertime. What gets our bees through the winter? It's those fat bodies. So it's actually adding up now. We're getting a better handle on what exactly is going on with the bees and the mites. The other thing they do, while they're feeding on those fat bodies in the honeybees, they're vectoring in viruses. We have a lot more viruses in our honeybees than we used to, because those mites are serving as a vector to get those into the bees. Remember, our bees have an exoskeleton. They don't have skin like we do. When they get pierced by a mite, that never heals. And we have different terms for the mites. We call a female mite that's on a nurse bee walking around the colony, we call that a phoretic mite. And a mite that's down in a cell we call a foundress mite. It's the same mite, it just depends where she is that particular time. So we just a terminology we use. The way it works is, when a female mite exits a cell when that larva hatches, or pupates and hatches, that female mite has already mated. She mates once in her life, and when she exits that cell, she's already mated, and she finds her way around onto a bee, usually a nurse bee, and she'll maybe pierce her exoskeleton, feed off of her, and then she'll wait until she comes to a cell that has a larva in it that's around 7 or 8 days old. Now everything in this is all science and biology, right? How do the bees know when to cap a cell? Do they look in and go, oh, that one's pretty big, I guess it's time to cap that over? Nope, it's all driven by pheromones. So these bees, or the worker bees, know that, okay, it's day 8, it's about time to cap this cell because I get that pheromone that this larva is ready to pupate. Well, the mites get that same thing. They say, oh, this one's going to be capped in another day or so. I think I'm going to go down in there. So the female mite enters that cell on day 8. She goes down to the bottom and she buries herself in the brood food at the bottom and she waits. And she waits for that cell to be capped. What she'll do is once it's capped, about 70 hours after she's entered that cell, she will emerge. Or sorry, that's not quite right. After that cell is capped, she emerges. 70 hours after entering that cell, she lays her first egg. That is a male mite. Every 30 hours after that, she will lay another egg, and those will be all females. All right? Each one of those females has to mate with her brother to become a fertilized female and be able to lay more, perpetuate the mites as they go forward. That's why the mites instinctively know to go to drone brood. That's what they prefer. Why? Because drones are 24-day cycle, right? And our worker bees are 21 days. She gets 3 extra days to raise more offspring. So she'll prefer drone brood, but she will go obviously into worker brood. And so she'll lay those eggs, and then what she'll do is she'll crawl up onto the side. She'll crawl up onto the side of that pupating larva, and she will chew a little feed port into the side of that larva, and then she'll defecate on the side of the cell. That gives her offspring, guides them up to where that feed port is. All right? So that's how she raises her brood. Every mature female mite, when that brood emerges, the adult or mature females will leave the cell. The male and the immature females stay behind in the cell. They never leave. They die. All right. So that's why she likes that drone brood, because she can raise more offspring. The mites are responsible for the vast majority of our colony losses. I've talked to a few people here, and I always get sort of the same story. And I know the answer before they tell me. I like to, not to embarrass anybody here, because we've all done this. I've done it. How many people here had colonies that were doing really well, maybe up to last month, or even if you talk about last year, you looked at them in February or March, they were doing great. April, they were all dead. Anybody ever have that? And they had plenty of food. We all have. And the first question I ask people is, when did you do your first mite treatment? And I get the same answer all the time. Oh, I did it in October, or I did it in November before I put them away. That was your first treatment? Yep. Too late. Way, way, way too late. These mites are feeding on the developing larvae that are going to pupate and emerge as our winter bees, right? Those bees, every bee that has a mite on it, its life expectancy drops anywhere from 20 to 40%. A bee with 2 mites on it, its lifespan will drop 40 to 80%. So what does that mean? We know that the physiology of our summer bees and winter bees are different. Our winter bees have more fat bodies in them, so they can get through that winter period. Our summer bees live what, 3-4 weeks? Our winter bees live 3-4 months. Well, when you're decreasing that lifespan by anywhere from 20 to 80%, Instead of that October bee that emerges coming through till April, now it's dying in February, right? So we see this classic symptom of, okay, my bees are great in February. I lifted, it was a warm day like today, lifted that cover, we looked in there, we saw the bees, great, I'm going to have a lot of colonies, I'm going to make a ton of honey this year. You come back in another month or so to start doing things and they're all dead. That classic sign. So I tell people, when you're going to treat for mites, you've got to do it early in the season. My own opinion, anytime after July, if your first treatment is after July, no matter what you do, it's a 50-50 chance of your bees living. Okay, some classic signs, right? Sudden collapse of a colony that was strong. It looked like a strong, healthy hive, especially early in the fall or late winter. Only weeks, could be weeks before they appeared really healthy, and you go in there and they're all just collapsed. Because again, they might not have all died from those mites, but enough died that the rest of the colony couldn't keep warm in the winter. They had a small cluster, and the rest of them maybe starved and just fell to the bottom as well and died. Bees walking around, seemingly doing nothing. You ever see bees that are just kind of walking around the entrance? They're not going in, they're not going out, or they just don't appear to be doing anything. Usually you look at your bees and you can tell they're doing something, right? Just kind of stumbling around. Deformed wing virus. Anybody ever here see that? Okay, if you haven't seen it, you haven't looked at your bees close enough. Because we almost always have it if we have a mite problem, especially a bad mite problem. Typically, I say if you're seeing a lot of deformed wing virus in a colony, that colony is pretty much dead already. You just don't know it. Cap cells that are punctured, a little hole in the center. The bees sense that larva has probably died, and they're in the process of removing it. So that's a sign, a lot of punctured cappings. I mean, there's other things that can cause that as well, but that's a typical mite one that you'll see. And Varroa might be observed on your bees during a hive inspection. Now, I tell people, don't rely on that. I cannot tell you. I wish, if I had $1 for every time someone said to me, I looked, but I didn't see any mites. I don't have any mites. I looked in the bees, I don't see any mites. I'd be retired. I wouldn't be here today. But, don't use that. You might see mites on bees. The one thing to remember is a lot of the mites are on the underside of the bees, so you're never going to see them. So those are some signs of mites. We're going to talk about testing, our mite levels and what to do. I think it's, really important that you do that. Now, I don't know how many colonies each of you have. Some of you might have 1, some of you might have 50 or, 100. I run a lot of colonies. I run 125 colonies, and I don't have time to test them all. But I test several hives in every one of my bee yards. And not so much to see if I have mites, because I know I have mites. I do it so I have a beforehand handle on what my mite counts are, averaging. You're going to get those colonies where they have very few mites. You're going to get a few colonies in the same apiary that have a lot of mites. But it doesn't matter. You're going to look for that average. Should I treat? And believe it or not, even if I don't have a booming mite problem in an apiary, I generally treat anyway, and I treat in July. So, because I know that even if I have a colony today that doesn't have a lot of mites, a colony over there, or my neighbor's colony that has a lot of mites, if his absconds from his hive, because it's got so many mites, it might end up in mine. So I generally have my mite treatment program going anyway. I use the before and after as a diagnostic to tell if the treatment I used worked. All right, so during the season, it's important to constantly monitor your mite count. It doesn't take a lot of time, and again, you don't have to do a lot of colonies. You can do 1 or 2 in an apiary. Things to remember during the mite season. When is the mite season? Anytime the bees are raising a brood. That population of your mites is going to double about every 30 days. Right? The next point is a really important one. For every 1 mite that we get in a mite count, depending on the time of year, there's probably 5 to 10 undercappings. So, you know, you could say, oh, I only found, you know, 3 mites or 5 mites per 100 bees. Well, if your colony at that point in time has 50,000 honeybees in it, do the math, right? I'm not good at math, so I won't try to do it in my head, but that's a lot of mites. So, you know, that's the thing that people don't remember, is that the majority of your mites at any one time are under cappings. And we have a lot of different diagnostic tests to determine if we have mites, what our mite load is, and then if we need to treat or not. Now, when mites first came around, they started killing our hives, and we came out with treatments for them, and we had recommendations on when, you know, what your mite count should be, when you should treat. And that threshold that way back when was, I think, was over 10 mites per 100 bees was fine. Now with all the viruses and whatnot that they're vectoring in, it seems like that threshold has dropped consistently. And right now it's, the recommendation is if you have 3 mites per 100 bees, you better treat. And it depends, of course, on the time of year, like as with everything else. I mean, 3 mites per 100 bees in late November is not a terrible thing because there's no brood. They're not raising a lot of brood. 3 mites per 100 bees in the beginning of, the end of March, beginning of April is a problem. It's going to be a problem at some point. Okay, so we'll talk about a few of the different treatment methods, or diagnostic methods, I should say. Sticky board. Anybody here use that sticky board? Yeah, a lot of people. I have to tell people, you know what? If you want to use that to see if you have mites, instead of spending the money on that, come and see me and just say, do I have mites? And I'll say, yes, you do. This used to be a very good diagnostic when we could tolerate those higher thresholds of mites, because it told you whether you had a lot of mites or a few. The problem with this is it's very inconsistent, because as the bees groom and depending on temperatures and whatnot, that mite fall off the bees varies. You could put that in today and get very few mite count, take it out, say I'm good, put it in that same hive a week later and it'll be covered with mites. So to me that's not a very good thing. It's easy to do, and if you want to do it, knock yourself out, go ahead. But I certainly don't use that as my diagnostic anymore. You know, I assume I have mites. You know, I know I have mites. To me, that all this does is tells me that I have mites. It doesn't do anything else for me. The one time you may want to put one in is when you're doing a treatment. You could slide that in when you put your mite treatment in, and then you should see a lot of mite drops. That might tell you, give you one indication that your treatment you used was very effective. That would be one of the only reasons I would use that. The cap drone scratch, that's a good one too. That one I like. And that again, it's just gives me an idea. Do I need to really start doing more testing or think about treating now? You know, come springtime, we're going to have, you know, our queen is going to start laying a lot of drone brood, right? Because they want to be able to raise and mate queens in that April, May, June, July period. So they're going to start raising a lot of drones. And most, you know, drones are what? They're male bees, so they're fairly useless, right? Men are useless. So you can sacrifice some. I like to take a capping scratcher, slide it under some capped drone brood, lift it out, and take a look. You can see right there's a mite on one of those. If you pulled something out like that, and I saw one mite, actually there's 2 on that one that I could see, actually 3, that would tell me to do some further diagnostics. If I've gone in and done several, and I saw, you know, maybe no mites early in the spring in that hive, I knew I was good for a while, so I didn't go any further with the diagnostics. But if I did that and I saw a fair number of mites on there, 3, 4, 5 mites on that much drone brood, I would certainly go to my next step of investigation. Ether roll. This was a popular one a while back. A lot of people still use it, but I don't use it anymore. It involves taking some bees, putting them in a jar like that, and spraying them down with a little bit of ether. The bees aren't crazy about it. It tends to kill them. The pros is pretty accurate and it's really easy. The con is it kills your bees. I know there's a lot of people that are hesitant to kill bees when they do their mite testing. I will tell you, the bees do it all the time, right? They sting us, they die. Why do they do that? They're sacrificing a few for the good of the colony. And I say that's what we're doing. We're sacrificing a few for the good of the colony. So I don't mind killing some bees when I do my mite test. And the most accurate mite tests kill the bees. With this one, we take our bees, we put them in that jar, spray some ether with them and kind of shake them around a bit, then we kind of roll it. What happens is the mites get dislodged. They'll stick to the side of that jar, and you can kind of roll it and count them. Like I said, that was a good test a few years back. Now we have better, easier ways, I believe. So I don't use this one anymore, but in a pinch you can. Sugar roll, for those of you pacifists that don't want to kill your bees, sugar roll's a good one. You're just giving them a little bath in confectioner sugar. They like that. Everybody's happy in the end. It's fine. We're going to take our bees. We're going to put them into a jar. We're going to put powdered sugar in there. The jar has a screen top, like I think it's number 8 mesh wire cloth, hardware cloth, number 8. So we put that over the top. Use a 2-piece canning jar, right? You replace the inner lid with that hardware cloth, and you just put the bees in there. You can put the powdered sugar through the top of the screen. You give it a good shake, 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 shake. Shake it enough. You're going to give the bees a little bit of a headache, but they're going to live, so it's okay. Shake it up, shake it up, shake it up. Set it out for in a warm area in the sun. Come back, shake it some more, because it takes a little bit to dislodge those bees when you're using the sugar. And then you take it and you turn it upside down like that. Usually I do it over some sort of light white surface. Shake it all out, shake it good, and you'll have that powdered sugar there. You may see some mites in there, in the sugar. Take a spray bottle with some water and spray that sugar to kind of liquefy it so you can see all the mites, and you do your count, right? That'll give you, that's probably, the second most accurate count you can do for mites, in my opinion. I don't use it very much anymore. It's easy to do, but to me it's a little too time consuming, so I don't use it anymore. I tend to do the next one. This one by far is my favorite one. Can you, the young fellow there, can you run and get that jar you were looking at? Tell Andrew just Frank wants it. Thank you. To me, this one's the best one to do. It's easy. It's again, you can do this cheaply. You can get any canning jar you have, and what we're going to do is we're going to put our bees in there, and we're going to put some alcohol in there. Don't use your best scotch or vodka. Save that for when you invite me over. Thank you.
You're welcome.
Rubbing alcohol, anything like that. We're going to take it, we're going to pour it on the bees like that. And it's definitely going to kill our bees, but it's going to separate those mites from the bees. If you're using just a canning jar, you just shake it around like this for a while. This is something that the makers of Apivar came out with this year. And I've used it and I actually like it. You know, I'm usually the cheapest one. I don't buy anything. But I've actually got one of these and I kind of like it now. It's a two-piece container. Put that in the top. It's got holes in it. We put our bees in there. We dump the alcohol in on them. We put the lid on. We shake it up. Swirl it around. The mites go to the bottom. We could take this out with the bees, and now all we've got is alcohol with the mites in it. We could dump it in something else. We could count them in there. Very, very easy to get a very accurate mite count. So again, I really like this. Gives you, like I said, a very accurate mite count, easy to do, and it's very quick. By the way, if anybody has any questions, feel free to ask them while I'm speaking. So we want to, no matter what test we do, we want to interpret our results, okay? First thing we need to do is we want to get, an accurate count. How many mites do we have? Well, if I just dump a whole bunch of bees in there and I do this shake and I get mites, okay, I got mites. Well, we know that, but how many do I need to treat? So we kind of want to keep track of what we're doing. Number one, we want to know how many bees approximately. We want to know how many bees we're getting. Quarter cup of bees is about 200 bees. Some people say to use a half cup, which they say is 300 to 400. I tend to use quarter cups of bees, right? So that's the first thing that we need to do. Second thing we need to do is we need to get the right bees. Common mistake people make, they don't get the right bees. They'll take it from anywhere. We want to get those nurse bees that are feeding that mature larva. Those are the bees we want. Those are the ones that the mites are on. Mites prefer to get on the adult bees, they prefer to get on nurse bees because the nurse bees are going to take them to that cell that's going to get capped, right? So those are the bees we want. So when we're in our colony, we're going to pull a frame out of there. We're going to look for open, mature brood that's about ready to be capped. That's the best place to get it, and we're going to look for the bees on that. Now, I will caution you to either make sure your queen is not on that frame, find her if you can. If you can't, just you look it over, have your friend look it over, take your time looking it over to make sure you don't get your queen, because we know this is going to kill her. So we don't want to do that. We want to get the mites, not her. So we're going to take that, and you take a little plastic, I always call them those little gift plastic buckets they give you when you're in the hospital. Here, take this home if you get sick at home, you know, what do they call them, puke bins, I think. Tupperware containers. I take one of those out in the apiary with me, and I take that frame when I've looked it all over, I shake those bees down into that. I take it, tap it down on its corner where I can scoop that quarter cup of bees and put it right in here, right? So now I know I've got my 200 bees in here. And this is where you have to know, just remember your math, because we want to know how many mites per 100 bees we have. So I've got 200 bees now in this container. I'm going to put my alcohol in here and I'm going to shake it up. And I'm going to count my mites. I'm going to count my mites. And, okay, so if I want, I'm sure I got a lot of smart people in here. If I have 200 bees and I want to know how many mites per 100 I have, what should I divide by? 2, there we go. So whatever amount of 100 bees you take for your sample, just divide it by that number. You got 500 bees, you divide by 5 at your final number. So if I did 200 bees, I came out with 8 mites, right? I did my division, I've got 4 mites per 100 bees. I better treat, right? So that's really, really important, really important. And when we're done treating, now our treatments that we're going to talk about, they're all different. Some are, you know, just a few days, 5 days, a week. Some of them are 42 days. At the end of our treatment, whatever hives, you know, you tested on, go back to that same hive and test it again when you're done. Because there are, like I said, there are treatments that sometimes, based on temperature and other factors, don't work. They're not effective. They don't do the job they're supposed to do. Don't believe just because you treated it all worked. Go back and do that diagnostic, right? Yes?
So why not just treat for prevention?
Pardon?
Why do you have to go through all this trouble of seeing, you know, how many mites per bee there are? Why not just treat for prevention?
You can do preventative treatment, just treat for mites if you want to. But the question then is, again, did it work? Did you have 5 mites beforehand? You did your treatment and you still have 5 mites per 100 bees. It's like I tell people, you know, do you have to do this diagnostic before and after? No, you don't have to. But when you go to the doctor, when you say, I feel sick, and he goes, oh, here's some antibiotics, good luck. No, he wants to do a couple of tests beforehand and a couple after to see if his treatment was effective. Someone else, yes?
Yeah, aren't you selecting for resistant to the treatment if you treat when you don't need to?
When you treat, I'm sorry?
If we treat when we don't need to, aren't we selecting for mice that are resistant?
If you're selecting for resistant mites, it depends. When you mean like raising your own bees to be mite resistant?
No, I mean when we treat unnecessarily, the mice that survive are the ones that resist.
If you treat unnecessarily, the mites that survive become stronger and more resistant to things. Well, I would have said that was true back in the early days of mites when we had check mite and apistan. They were very hard chemicals. People didn't use them according to directions. They left them in too long. So there was a little bit of residual still, but not enough to kill the mites, and the mites developed their resistance. The vast majority of our treatments today run along the lines of a more natural treatment that There's organic treatments that the mites are going to have trouble building resistance to, like formic acid, like oxalic acid. And there's other ones that they may be able to. The chemical kind are the ones they can develop resistance to, like maybe an apivar. So far, apivar has not shown that, only because it breaks down so quickly. Whereas the coumaphos and fluvalinate, those didn't break down quickly. And they were getting those very, very small doses, and it built resistance in the mites. We're not having that issue at the moment, and that's, I'll talk a little bit more about that when we talk about treatments. But yeah, I think that if you think you're going to raise bees that are resistant to mites, you're in the wrong hobby. We are not going to do that. That's like you saying, hey, you know, I'm getting a Chihuahua. I'm going to teach my Chihuahua that to live in the Arctic. So I'm just going to take him up to the Arctic and put him out in the snow, and he'll be fine. It doesn't work that way. It takes years and years and years, right? And it takes selective breeding. That's what we have these scientists and bee breeders doing. We, as hobbyists, have very difficult time. There are a few guys around that are treatment-free. You talk Kirk Webster up in Vermont. He's pretty treatment-free, but he loses a lot of bees, and he's okay with that. He's trying to breed it into his colonies, you know, and he's got a lot of colonies. We as hobbyists can't do that. We just can't. You can buy bees that are hygienic somewhat, but any bee that any queen you buy that's a hygienic queen, I don't think any of them say, hey, here's a hygienic queen, you can stop treating. You don't have to treat anymore. You may not have to treat as often or as much, but you still have to treat. If we had bees that we could raise that were treatment-free, every commercial beekeeper in the country would be running those queens, because that's their biggest problem now. They're losing, you know, this year was a horrible year. You know, I've heard on the whole East Coast, I have commercial beekeepers that have lost 50% of their colonies. You know, so, you know, if they could be treatment-free, they would be. So we do have to treat, you know, we do. We're not in the business of being treatment-free, and it's a disservice to your neighbors, you know. Here, especially here in New Jersey, I know you guys, New Jersey is a little more populous than some of the other states, so, and a lot of hobbyists in New Jersey. So if your neighbor a mile away decides they're not going to treat for mites, which a lot of people do, they say, I'm not going to treat, I'm just going to be a natural beekeeper. And then they say, well, I was doing really well, then my bees disappeared. Really? Well, where'd those bees go? When they abscond from a colony, they get overrun with mites and they abscond, they're going to look for someplace else to go. They might end up in your colony. And you've taken care of your mites and treated your bees, and suddenly now you've got a hive full of bees. So you also have to think about this treatment as being responsible not only for your bees but your neighbor's bees. And it was, I think again, it was Dennis Van Engelstorp and his crew did an experiment where they had an apiary a mile away where they did not treat, and they had an apiary where they were doing all the treatments. And their mite levels in that apiary where they were doing the treatment were all below that threshold. And they were testing them, I think, weekly. One week they tested and the hive had something like 1 or 2 mites per 100 bees. They came back a week later, it was like 30 mites per 100 bees. And they went to the other apiary, those bees were gone, that they weren't treating. They were gone, they were over here. I think they even marked them or something. So you've got to be responsible and treat. You know, it's just the right thing to do. And if you don't, it's going to be frustrating because you will lose your bees. You know, like I said, I lost lots and lots of bees every single year. You know, no, I almost gave it up. And it wasn't until I got a handle on mites plus everything else in beekeeping that I became a lot more successful. Okay, so we're going to talk about how we treat. It's going to depend on the time of the year and the state of our hives, right? Is it early spring? Is it summer? Is it fall? Do we have honey supers on? Are we about to super our hives up? Did we just pull them off? And I'm going to talk about, the different treatments, some of them. And again, I'm not going to tell you what treatment to use. I'll tell you what I do. What I do works for me. Maybe it doesn't work for you. Maybe you have to do something different. But it's up to you to pick something. The 2 things I'll stress are, number 1, pick something. And number 2, do that treatment, your first treatment, no later than July. We want our bees to be relatively mite-free in that August, September, October time frame when they're raising those winter bees. So the first I'm going to talk about is probably to me, the most effective treatment, but not. It's a chemical treatment. It's Apivar. The chemical name is Amitraz. I will tell you, this is what almost every commercial beekeeper uses. A lot of them are using it off-label because in that pack, Apivar, very expensive. I think that pack, that 10 pack is probably, I don't know, 30 bucks or more, I don't know. But it's a very expensive treatment. It's a very long treatment. It's 42 days, but it's a very effective treatment as well. It cannot be used with honey supers on the hive. So again, this is where you do your planning. In my case, I like it because it's a contact. So far, there have been a few little reports here and there of resistance, but it hasn't been confirmed because, you know, it always comes into question whether they actually followed the directions. I found people that were treating, you know, these slide down like that through the frames. I've had people tell me they just slid them in the entrance. And then they said, I think the bees are resistant to it, it didn't work. Well, you didn't use it according to directions. Really important to use everything you use, anything you use it to directions. They're plastic strips impregnated with that Amitraz. The bees walk on and make contact with them, and it gets on the mites, kills the mites. I like it. I put it I put 2 strips right in the brood nest, and in the box above it, I alternate them and put them on different frames. So usually I have total, in that 2 box seam of frames, I have the 4 center frames where the majority of brood is being raised. I have that covered with the strips, right? So no matter which of those seams that a nurse bee goes up, they're going to be exposed to that Amitraz, and it's going to kill those mites because those mites are on those nurse bees. Very, very effective treatment. Again, you can't have honey supers on, and it's a 42-day treatment, long period. In my beekeeping, I know, again, you should know everything about your beekeeping, everything that affects your beekeeping. I know my honey flow. In northeastern Pennsylvania, where I live, my honey flow starts with dandelion bloom, and it's over by about the 4th of July, depending on if we have basswood that year or not. It's usually over by that 4th of July. As soon as that flow is over, I pull my honey supers. I'm a guy who I pull honey throughout the spring season when I want to get my varietals. Sometimes there's no varietal, but I pull them all by that 4th of July-ish period. So now I've got no honey supers on my colony. I can treat with anything I want. I treat with Apivar. And for me, that's my first treatment of the year because the mites start out very low in the springtime. They need brood to reproduce. No brood, they're not reproducing the mites. And it takes them a while to get going. It starts out very low in the spring, it starts climbing, and it peaks in August, right? So I know, beginning of July, it has not peaked yet. I never want my mites to peak in that August time frame without a treatment. That's where our bees are going like this, and our mites are following it. By the time we get to August, our bees have started dropping in reproduction, but the mites are still going. You cross that without having done a treatment, your bees are in trouble. You're probably going to lose your bees. That's why it's very important to treat earlier. So I pull my honey supers in the beginning of July. I put my Apivar in. I pull the honey supers off, put them on my pallets, do my treatment right then, close the hive up, and I let it go for 42 days. When I come back, my fall flow starts in the middle of August, and I come back, I remove those strips a week ahead, I put my fall flow honey supers back on, and there I go. Yes?
Do you recommend also treating like in the early spring?
I don't recommend treating in the spring, but I don't recommend against it either. It's a personal thing. My mite treatment program of treating once a year with Apivar works for me. Now, if you were using something maybe, which we'll get to, something that's maybe not quite as effective as this, because this is, like I said, it's a chemical. You don't want to use this. You want to go something with one of the more natural ones. You may want to do one in the spring to knock them down, because the softer the treatment is, the less effective it is. This is very effective because it's one of the harder treatments. Now, the one reason why I'm okay with using this, I would never use, you know, hopefully no one's listening, but I wouldn't use the Coumaphosphorvalonate, the check mite, and the apistan. I would never ever put those in my hives. Those chemicals break down very slowly, they get absorbed into the wax, and they never go away. That's why most of the wax in this country is polluted, the beeswax, because it still has those chemicals in them, and those products are still being used. There's the one of them, check mite's very effective on hive beetles, so they still use it. Yes?
You said you use 2 strips per box.
Yep, brood box.
Yes. That's independent of how strong the hive is?
Yes. The question was, I said I use 2 strips per brood box. I do. Why? That's what the manufacturer tells you to do. Always follow directions. And doesn't matter the strength of the hive. Now, if it's a nuke I'm treating, I put 1 strip. It's basically 1 strip for every 5 frames. So, and yes?
And not to nitpick, but if you run mediums, would you still use the same number of strips?
Yep. If you run mediums, would you use the same number of strips? Yes, I would. Yes. A lot of people run mediums. I have some mediums. I do it the same. Yes?
Yeah, you say you pull your honey supers in July, you harvest them. Can you put the harvest honey supers back on top of the inner cover at that same time?
You could, but I wouldn't. I would, if you mean to let the bees clean them out, I would set those out for a day or 2 away from my apiary, let the bees come and rob them out. If I were using apivar, that is, I wouldn't, because they would track that apivar up in there. Now, I said about apivar, it's a long treatment, 42 days. Those plastic strips are impregnated with that amitraz, and it slowly comes out of that plastic. That's why it's so long. But the good part about amitraz is it has a very quick half-life. I think it breaks down in 6 hours after it hits oxygen. Oxygen causes it to break down, so it does not last. So, you know, even they tell you to wait a week, I think, before you put your honey supers back on when you're done with this treatment. But all the tests show that within a day, there's almost no trace left. Amitraz breaks down into 2 metabolites that are, so far, they say is harmless to everything. So, but that's mine. And one of the reasons why I use that is, you know, number one, I have a full-time job. I'm busy and travel a lot. And I run a lot of colonies. I run 125 colonies at the moment, and I just don't have the time to get to all of them to do another treatment, a softer treatment. When I retire, I will start using some of the other ones because I'll have more time to spend in my apiary. So, the next one is Mightaway quick strips. They just reformulated that. What's it called? I saw somebody.
It's a quick question.
Yeah.
About equipment. If you've lost hives to Varroa, is there a treatment for the equipment, or can you reuse the equipment?
The question is, if you lost a hive to Varroa, can you reuse the equipment, or do you have to do a treatment on it? It's perfectly fine to reuse everything as is. You don't have to do a thing, because what you have to remember is no brood, no mites. Brood's all dead, the mites are all dead. So, yep, they can't live in the hive without bees. Yes?
If you don't use all your apivar strips in 1 year, can you use it?
No. The question on the apivar strips, if you don't use them all in 1 year, can you use the remainder? No. Somebody asked me that in the sales area earlier. That's the one downside to apivar, in my opinion, is the way they package it. For the average hobby beekeeper, they don't package it in numbers that make sense. Once you open that packet and it hits oxygen, the degradation starts, and there's nothing you can do. You can put it in those vacuum sealers, everything. No, it's no good. It won't be effective. And the worst thing you could do is put an ineffective treatment in, thinking it's effective. So if I think there are 10 counts in there, and you need 4 strips for if you have 1 hive, you need 4 strips. Well, the best thing to do is find a friend that has 1 hive, and you split the pack. At least you're not wasting that much money. So, yes? No, if there's no brood in it, I might not. I probably would, just to make sure that the bees all get exposed to it, because those bees that are in the bottom box are eventually going to go up to the top. So I would, if I had 2 boxes, I'd use the 4 strips. You know, there's times that I have 1 brood box, you know, brand new hive, and I'll just put 2 strips on it. Yes?
If you have an unopened package from the year prior, can you still use that?
Yeah, I think so. Look at the expiration date. I think it's good for a year or 2 from the date they put on there. If it's unopened, it should be okay, but I wouldn't keep it forever because even though they're in those sealed packets, oxygen, finds a way into everything. Yes?
We've done that, and it doesn't save.
It doesn't save, right? Yeah.
If you open them, the strips are even degraded.
Yeah, so it's better to, what are you talking about? You talking about the Mightaway quick strips or the Apivar? Okay, yeah.
If we throw it away.
Yeah, Apivar, buy it when you're going to use it. So it's very expensive stuff, don't waste it. Yes?
Hi, are you using Apivar just once a year?
Yeah, in my treatment, I am. Because I'm finding, and like I said, I do things, my beekeeping, which may not fit your beekeeping, because part of my beekeeping is that come that June through the first week of July, I'm also splitting, almost not splitting, but pulling frames of brood out of all my hives to make my nukes to overwinter. So I'm doing a break in the brood cycle almost there as well, because a lot of times I'll take the original queen and move her to the nuke and put a new caged queen in the original hive. That gives me a break in that brood rearing cycle. So yes, mine's a bit different, but that works for me. And again, that's where that testing comes in. You do your testing, you do that treatment in June, July, well then you might want to do a test, depending on what your treatment is sometime in August, and see what your mite count looks like, and you might just have to, they're slowing their brood rearing down come August, September, you might be able to use a lighter treatment. One of these, Mightaway quick strips. Mightaway quick strips, that's one of those natural treatments. It's formic acid. Formic acid occurs naturally in honey. It's there. So it's not a chemical, it's not, the bees aren't adverse to it. They don't like it because it's very strong smelling. You'll see them come pouring out of the hive and, bearding out in the front when you first put them in. It's a 7-day treatment because over the course of the 7 days, that formic acid gases off of those pads. When you put it in, it's an acid, be careful. Wear nitrile gloves so you'll burn your hands. Put it in just like that, between your 2 brood boxes, 2 strips. One of the pros is you could put it on when you have your honey supers on. So if you tested your hive and you found you had mites in June, right in the middle of the honey flow, you could say, and throw these on. It's fine. The cons is it's an acid, it'll burn you, and you got to wear nitrile gloves, and you got to kind of avoid breathing the vapors. There are reports, and it's very temperature sensitive. This is a fumigant, and it has to get spread through the whole hive, and the bees have to fan it around. It works by the bees actually fanning. They don't like it, and they'll fan. So it's very temperature controlled. If it's cold and the bees are in cluster, right? Bees start going into cluster at what, 57 degrees or something? If it's in the 40s and they're in total cluster, well, they're not going to fan that around. It's going to be very ineffective. On the other hand, if the temperatures are high in the 80s, mid to high 80s or higher, it gases off so fast that it kills bees. It'll kill brood and it'll affect your queen. A lot of hives go queenless. They think some of the thoughts are that it masks that queen's pheromone and the bees kill their own queen. But it's well documented that your hive could go queenless. Generally they'll raise a new queen, but still you don't want to lose that time period. So it's a good one to use if you have your honey supers on. It's during the honey flow. I like it. I've used it, but like I said, I've had spotty results with it. Yes?
I've heard that putting Mightaway quick strips on while your supers are on will virtually stop the influx of honey.
The question was, he said he's heard that if you put it on, Mightaway quick strips on when your honey supers are on, the bees will stop foraging honey, putting honey up in the honey supers. Very possible, because they're busy trying to fan those fumes out of there. But the good part is it's a very short, it's that 7-day period, and really the majority of that stuff gases off in the first 2 days. So it might slow them down, you might be correct, so you might want to think about that. When you're in the big flow, then you might want to wait a little bit. I don't know how your flow goes here in Jersey. In PA in my spring, we call the minor flow in May, it drops a little bit toward the end of May, then we have the major flow in June. So if I were using that, I'd probably time it around that. Yes?
One of the things that I'm using Mightaway quick strips is if you don't put the queen excluder up, you're going to force the queen up into your supers too.
Yeah, and again, different beekeeping. He said if you don't put a queen excluder on, you have your honey supers up, you're going to push that queen up. With me, I don't care normally, depending on the time of year. I don't always run queen excluders, a whole different topic, but to me, it's all management. I don't mind my queen going up into that honey super sometimes, especially if it's a brand new box with new foundation and they're just starting to draw it out. I let her go up there and lay a few eggs, but I know come the beginning of June, I'll make sure she's down, and then I'll put that queen excluder on. That brood will all pupate, hatch out by the end of the honey flow. No brood in my honey supers. So it's different, just different management. Hop Guard, this is one that came out in the last few years. Hop Guard is made from the beta acids of the hops plant. Again, it's a contact, the bees don't like it. You can use it with honey supers on, it's non-toxic to humans. It might irritate your skin if you put it on with your bare hands. And the cons are the mites have to make contact with it. It's pretty short-lived because, like, you know, bees, we've all seen stuff in the hives that we put in there, newspaper to join the hives, whatever. The bees don't like something, they remove it from the hive. That's how this works. And it's a multiple treatment. You have to treat I think it's 3 treatments over 2 weeks apart or something like that. Maybe it's only 2 treatments 2 weeks apart. I have not used it. Again, only because I don't have time to go back to my hives when I'm doing this. If I had the time, I might try this. Some people like it. I have some fairly good-sized beekeepers that this is all they use. Other ones say, no, they don't like it. So again, but it's a very natural treatment, really natural, not a chemical. Thymol treatments, we don't sell these. I think it's a day damp product. But it's made from the thyme plant, and from what I've heard, it's fairly effective. It's, again, a natural treatment. And the more natural they are, the less effective they are, but they work. And the bees, these again, they're temperature sensitive. They gas off that thymol, which is a very strong smell, and the bees don't like it, so they'll try to remove all that from the hive. And part of that, as they're removing it, they're getting it on themselves, they're getting it on the mites. It'll kill mites. It's probably not going to be your only treatment, so you'd have to really monitor your hives when you're using this and afterward. And retreatment is almost always necessary with this. It's not a one and done. It's either this several times or this plus something else later. Oxalic acid. Anybody here have those vaporizers, oxalic vaporizers, or do the oxalic drip? I'm hearing different things. Some people think it's fantastic and it works great. Other people say, I haven't seen much difference with it. I believe it probably does work to some extent, and it's probably a pretty good treatment. And the good part is, you know, you can, I don't believe you can have honey supers on when you do it, but the dribble I don't think I would ever use. It's too difficult to get right, in my opinion. You got to do about 5 milliliters per seam for it to be effective. And if you put too much on, you'll kill bees. You can only do the dribble treatment apparently once, or if you do it a second time right away, you'll kill bees. I don't know how true that is, but that's what I've been told. The vaporizing seems to be very good. Again, I don't do that because it's too time consuming for me. It takes probably start to finish, what? Anybody that's done it, what, 5 minutes per colony maybe, right? Yeah, and so if you got a lot of colonies, it's not very effective. Now they're coming out with some new application methods. There's a gun that they came out with that you load up and it's seconds in that hive, switch the cap, fill it, go to the next hive. That would work for me, but you need a power source in your apiaries. So I haven't done it yet, but I'm going to. I plan to, and I want to test it. This is one that, the good part about this is you can do it, you know, anytime almost. The one thing you have to remember, it's only effective in a broodless colony because it doesn't do anything for the mites under cappings. I did neglect to tell you that on the Mightaway quick strips, it's the one product that will kill mites under cappings. As the bees fan that around, it actually penetrates the cappings, or they're not porous, but oxygen can pass through the wax cappings on cells. That's why that one works for mites under cappings. But this one does not, so it's mostly ineffective in a colony that has a lot of brood in it. It's not very effective at all. So this would be one of those, if you're going to, a lot of people do this, they have multiple mite treatments through the season. They'll use this in the springtime because there's very little brood. So they'll use this in the springtime to get a fairly good mite knockdown. Summer, they'll treat with something else. And then fall, they'll do this right in the fall to knock down any mites that are on bees. Okay? So it's not a bad treatment. Be very, very careful with it. Those vapors, what happens with those vaporizers, it actually sublimates. It goes directly from a solid to a gas. It does not go to a liquid. So it's sublimation. It goes solid to a gas, and then it goes from a gas right back to a solid. And the bees and the mites, it gets on, supposedly burns the pads of the mites or something. But think about, and when it's going to that gas, you'll see anybody doing it, the hive billows with that, right? You'll see it coming out the top of the hive. If you breathe that gas in, guess where it's going to go to a solid? It's going to go to a solid in your lungs. Not good for you. Very, very bad. So don't ever use this without what do they call it? An acid vapor respirator. Yeah, no hive of bees is worth your health. So, but again, I'm not going to say anything bad about it because I think it's a valid treatment. So, in summary, that's pretty much all I want to say. You know, as a beekeeper, you know, again, it's not as easy as it used to be. So, as a beekeeper, you got to become familiar with how all this biology works, right? If I do a treatment my only treatment is a treatment that's ineffective in a colony with brood in it, and I do it in a time when his colony's full of brood, well, I've wasted my time, I've wasted my money, and my bees died anyway. If I do my first treatment of the year in October, November, I've wasted my time, wasted my money, my bees are going to die. If I did my only treatment in March, I'm probably wasting my time too. You need to really think about everything you're doing and come up with a plan that works for you. What works for me may not work for you. What works for her may not work for him. It's what you're comfortable with, what you want to do. And again, part of that whole thing is monitoring to see if what you're doing is working. If you get your Varroa under control, you will be a much more successful beekeeper. Will you still lose bees in the wintertime? Probably. I'm going to talk about that, I think, later today or tomorrow. They're all today, I think. Yeah, we'll talk about that today, sustainability in an apiary. But, you know, all of the stuff, every part of beekeeping, there's a little puzzle. There's a little piece of a puzzle. Whether it's, you know, Varroa treatment, nutrition, sustainability, all of that stuff. It's when you put all those pieces of the puzzle together that you're going to become successful. You get very good at one thing, it doesn't mean you're going to be a successful beekeeper. You have to be pretty good at all of them. And that's where you'll see a big difference, because this hobby of ours is so much fun, but it can be so frustrating. I mean, how many of you at one time or another said, I have no clue why the hell I'm doing this, right? You know. So, you know, we don't want to be like that. We want this to be a fun thing, hopefully not too expensive. A lot of these treatments are cheap. That oxalic acid treatment, extremely cheap. You can do those for almost pennies. So, you know, it's mostly the knowledge and putting it all together that is what makes us successful. And like I said, you know, you guys got great clubs in New Jersey. Go to your club meetings, go to conferences, read, be careful what you watch on the internet. You know, if you don't recognize the name like Mike Palmer or one of those guys, don't watch it. I've seen some of the most horrendous stuff on the internet, and you know, I stand in my store sometimes and someone will come in and they got bees from me last year, they started keeping bees. They'll come into the store and I'm talking to somebody and another customer will come in and the guy that started keeping bees last year is telling the new beekeeper how to keep bees. I want to go, oh my God. So make sure you know who you're getting your info from. I say if you haven't been keeping, person hasn't had bees for 5 years and lost all their colonies at least once and now they're successful, don't listen to them. Walk away. So, and I think there's some obviously great people in this room. I know a lot of them that are very good beekeepers. And of course you're getting, I think the king is coming tomorrow. Grant Stiles going to be here tomorrow, I think. Yeah, Grant, if anybody, those of you that know Grant, I don't have to say anything else. Those of you who don't know Grant, go meet him. You'll walk away a changed person. So that's all I have. If you've got any questions now or later, I'll be happy to answer them. Yes?
Yeah, the silic acid, is that with or without honey supers?
It's without honey supers. Yeah. I think after you've done the treatment, you can put them right back on. You know, if you took them off, treated, put them right back on, I think you can do that. But you don't want them on while you're treating. I think I'm correct in that, correct? But I think they're doing testing right now, as a matter of fact, of leaving honey supers on. Jennifer Berry down in Georgia was doing some testing of treating with honey supers on to see, you know, if there's any residual in the honey and whatnot. Because oxalic acid is a very natural acid as well. It's in rhubarb and some other plants. So, spinach, yep.
It's also a toxic.
It's a toxic, but we eat rhubarb. Hey, just put some strawberries in it, sweetens it up, we're good. Yeah. Yes?
Can you overtreat with Acabar?
Can what?
Overtreat with Acabar.
The only, yeah, you can. You can overtreat with Apivar, certainly. I mean, it calls for 2 strips per brood box for a reason, right? I mean, if you overtreat, will it hurt anything? No. All it'll hurt is your wallet. Yes?
With the time that, you know, you do the treatment of the Mightaway strips, or temperature sensitive.
Right.
With our summers seem to be getting a little bit warmer now. Are they working on anything new or different?
Yes. The question was the Mightaway quick strips with our summers getting warmer and whatnot, and that being very temperature dependent or sensitive. Are they working on anything new? Yeah, this presentation was from like a year or 2 ago, and I have Mightaway quick strips on there. Somebody in here, I saw somebody buy some out there. What are they called now? Formic Pro, thank you. It went out of my head there. Their reformulation is called Formic Pro, and you can treat differently. They used to tell you, don't deviate from this treatment. 2 strips between the 2 brood boxes. Now they openly say, and the guys are running 1 strip instead of 2, and they weren't getting the knockdown and the effectiveness. Now with the Formic Pro, there's several different options on how to treat. I think it gases off a little more consistently. It doesn't gas off so much in the beginning and less at the end of the 7 days. I think they've got it so it's about the same every single day. And now they've said, okay, well, you can use 1 strip with the new formulation, 1 strip between the brood boxes, come back in a week later or 2 weeks later and do it again with 1 strip again. So you're effectively doing 2 treatments, you know. So I haven't tested the new one yet. I'm hopefully going to test it this summer in our bee yard at work and see how it works for me. And I'm going to test the temperature ranges and whatnot. So, yes?
Any work that you know of concentrating on interrupting the fertility of the mite?
They're always trying different things. The question was, are they trying to come up with something to interrupt the fertility of the mites? I don't think there's anything on the forefront coming, but they're certainly looking at things. How can they, you know, maybe render the male sterile, which they've done with other insect species and whatnot. But all of that stuff I believe is quite a ways away. I haven't heard of anything. So, there's a man with his arm up. Do you know anything about that?
No.
Oh, okay. He just looked he looked very energetic. I thought we had a scientist from the USDA here or something.
One more question.
What's that?
We can do one more question.
I can do one more question, and he was...
The native bee in America was what? Because we have Italian bees, Grenolian. Why don't we just get the Asian bee over here and that can handle the mite?
You saw how that worked out with the Japanese beetles, right? You saw how that bringing invasive species in worked out, right? No, the Asian honeybee probably wouldn't survive here, I don't think. I think it's a different subspecies. I don't know. There are reasons why we don't have them. I think I've heard them before. I don't remember, but we don't have, you know, there's reasons why.
I think I was just reading that their honey output is a lot less.
That's probably a big part of it, too, yeah. I mean, Apis mellifera makes the biggest colonies there are, right? So, unfortunately, I have to stop. But if you have questions, I'll be out there in the vendor area or walking around here. You're more than welcome to come grab me and ask me anything you like. Thank you very much.